Okay, well, first, thanks for David and Ryan for organizing this. Since this is like the von Vreeswijk seminar, I just also wanted to say a couple of words about Carl von Vreeswijk, because as you said, I did my PhD around Paris, and I had the privilege. I very briefly ventured in the world of recurrent network, and so I had a few chances to interact with Carl, and he was just a great guy, so generous about what he knows and so encouraging that I think it was a pleasure. I mean, it was also clear to me that recurrent network I was not smart enough to do recurrent network theory, but at least I learned that out of it. Okay, so today I'll talk about sensory processing and mostly talking about the retina, but my hope is that I will try to convey doesn't necessarily apply in principle, not just to the retina. So if you don't care about the retina, you can still bear with me, hopefully. And to start with, I just want to somehow make a broader comment about what we usually call reductionism in neuroscience. I mean, ideally, when we study mouse visual processing, we would like to understand how the mouse process visual information in a complex environment like this one. But usually the way we try to do this is by breaking down the visual scene into components. So usually we kind of more or less freeze the mouse, head fixed, and then we decompose this natural environment into components. So here, for example, Fourier components. And this is very well controlled, but of course the issue is that it's pretty far from the real ecological experience. But the hope there is that by studying how this is processed by the visual system, the mouse, you should be able to draw hypotheses that you can later on test in more complex environment. And this has been successful in many ways, but of course there are also some cases where it fails. And for that reasons, people have also developed approaches where, put that here, where they try to directly look at the animal in a complex environment. So like freely moving mouse and complex visual scene and all these things. But of course the issue with that approach on the other end is that you don't control things so well as that whenever you see a neuron responding to something, you don't know what that something is. And usually several things are changing at the same time, so you have the challenge of trying to really make causal inference about who is triggering what in what neuron. And of course there are ways to try to get around that, but basically what I will talk about today is one approach among many others to try to somewhat find a middle ground between these two cases. And I'm gonna work. Oops, sorry. I'm gonna work in the retina because I think that already in the retina we have that kind of problem. So this is the retina. So you probably know, all of you, that this is the thin piece of tissue at the back of your eye that is doing already many interesting nonlinear computation. It starts with photoreceptors that transduce light into electrical activity. Then it goes down through several layers of cells down to ganglion cells. And ganglion cells, the output of the retina, they are sending their spikes to the brain. And we know a lot about how ganglion cell process simple stimuli, like artificial stimuli like white noise, moving bars. We know about receptive fields, center and surround. But I think it's fair to say that we know much less about how they process complex natural scenes. I mean, people have tried various approaches to bridge the gap between the artificial stimuli and the natural one, but I think we're still not completely done with trying to understand what is the features that are extracted by ganglion cell from natural scenes. Just as an illustration of that, if you want to know what a single ganglion cell care about, so to say, let's say you're just recording that on ganglion cell. Well, the classical approach is that you will actually display several random noise-like patterns, so like checkerboard-like patterns, and you will. I'm gonna simplify things here, but you probably get the idea. You're gonna just count how many spikes is evoked by each of these patterns. And then you're gonna do a sort of a correlation between this pattern and the number of spikes. So you're gonna do a weighted average of all these patterns, and you're gonna obtain something like this. So it's what we call usually a spike trigger average, which is a good proxy for the receptive field of the cell. And in that case, it tells you that this ganglion cell is selective to light increase. So its response will increase when you increase light in this area. Okay. So this is bread and butter of sensory processing and of retinal studies. But of course, there is an issue here. And the issue is the following. So if you take that, that receptive field, which is kind of and you treat it as a filter. And basically, you can just add a few bells and whistles after this filter, but there's nothing very important here, such that you can just take an input, filter it with this filter, and then have a few nonlinearity here to just have some output spikes. Then you can use this as a predictor. And if you do that on the white noise I was showing to you earlier, it works pretty well. So here what you have, this is a paper from E.J. Shishlinsky group, but this is a point which I think is relatively well known in the retina. So here you're presenting the same sequence of random white noise several times. Each line is the same sequence repeated, and this is the response of one neuron. So you see you have very reproducible responses. And if you ask this model based on the receptive field to predict the responses, it actually does very well. Okay, so you see there is a pretty good similarity between the experiment and the model here. So it works quite well. The issue, though, is that now if you do that for the same neuron, but not trying to predict the response of the same neuron, same ganglion cell, to a natural movie, then it fails miserably. I mean, I think it's fair to say that there's a lot of differences between the experimental responses in black and the prediction in red. And this has been seen multiple times. So there's a lot of nonlinear processing happening, and for that reason, it's not easy to go from this model learn on the artificial stimulus and try to predict what happens for natural scene. Okay, so this is somehow our starting point. Several people have addressed this issue, but let me actually tell how we decided to talk about it. So first, see there's some question there. Okay, sorry. All right, so basically here our starting point was to actually take this hypothesis seriously. So you start from this receptive field here, and this is telling you, okay, you should increase light in this area. Well, if this is really true, of course, this is a bit of a straw man, but let's say if this is really true, if you take that hypothesis seriously, and if it still applies to natural scene, despite the failure of the ELN model here, then if you take a natural image, and let's say this cell is responding like this to this natural image, if really what this cell care only about local light increase in this area, then the best thing to do to increase the response of that cell to this image should be to somewhat increase light in this area. So obtain something like this, where I just have added to this image this blob, and this should be the best way to increase the response of that cell. Okay, so all right, this is an hypothesis we can test, and this is actually what we decided to test experimentally directly on natural images. And so for that, I'm going to say only a few things about the recording methods, but let's say we just recorded from various species, mostly mouse, but there's also a bit of axolotl in my slide. And the method we use is basically multi-electrode array. So you euthanize the animal, take the eye out, take the retina out, and flatten the retina against an array of electrodes. And from this, through extracellular recording and spike sorting, you can extract the activity of a couple of hundred cells. Although mostly what I will talk to you about is actually single cell work. Okay, so again, first we're gonna just record a neuron and present as a stimulus this reference image, okay, this natural image. And then we're gonna add to this reference image a little perturbation that takes the form of this kind of noise pattern. And we calibrated the amplitude of this noise pattern that we're gonna add to this image so that it slightly changes the response. Okay, so we're gonna obtain something like this. I have enhanced a bit the contrast of the pattern here, but the idea here is that by adding this pattern, we should slightly change the response of the cell. Okay, and then we've been doing that, keeping always the same reference image, but every time changing the perturbation. So if you want to think about it a bit more abstractly, what we're doing essentially is that this image is one point in the stimulus space. And what we are doing is that we are poking in the stimulus space by adding these different perturbations in various directions. So we're just exploring the neighborhood by doing various perturbations, applying them, and then we record the response of a given cell, and we see how these various perturbations are changing the response. So some of them increase it, some of them decrease it, etc. And now we can do the same thing as we were doing before. We can correlate the change in the response with the perturbation. And for this, you can just simply do a weighted average of the perturbation weighted by the number of spikes. And if you do that, you obtain, as before, a spike trigger average, sort of a local receptive field. But the difference here, we call it the local STA, because now this receptive field is somewhat attached to the image. I mean, remember, we only explored the local neighborhood of the stimulus space around the natural image. Okay, that's what we call it, this local spike trigger average. And one way to interpret it is that basically this spike trigger average tells you, okay, if your purpose is to increase the number of spikes, to increase the response of the cell to this image, well, then the best thing to do in that case is to increase light in this area. Okay, so here in that case, you're like, this is red, so this is positive. So basically, this is telling you, okay, starting from this image, you should increase light in this area. Okay. So now here are the kind of results we get with this. So these are just two cells, very classical cells. This is a very classical off cells from the axonal retina. This is a classical on cells from the mouse retina. This is the receptive field measured with the standard technique I described in the first place. And so this is off cell, this is on cell. And now here, what I'm showing to you is the result of the method I just described. So we measure, the local receptive field or the local spike trigger average when you use this as a reference image or when you use this as a reference image. So you have the two coupled here. And so far you see that everything seems to be relatively expected. So if you start from this image, the best thing to do to increase the response is to decrease light in this area. If you start from that image, more or less the same story. Yeah, you see a little displacement, but nothing very serious maybe. And you still should still decrease the light in this area. And for that, so this is all the same cell here. For that other cell, this on ganglion cell, now it's an on cell. And here, if you want to increase the response to this image, you should increase light in this area, and here too. This ellipse is basically the same everywhere. Here, this is just used as a visual landmark. Okay. All right. Now, the surprising result is that still looking at the same two cells, but changing the starting point, starting the image that you perturb, you can get very different answers. So here, if you start from this image now this perturbative analysis tells you that if you want to increase the response of the cell to this image, you should increase light in this area, despite the fact that this was an off cell. And same for that images too. And here for this on cell, well, you can find also some images where if you want to increase the response, now you have to decrease light in this area. And the same happens here. So depending on the image, your cell can change polarity. What I mean by polarity here is really on and off polarity. And this is, a bit surprising. We don't see that with every cell. There are some cells that are really, on or off, and this is carved in the stone. They stay on or off forever. And we don't obtain this kind of effect, but still a bunch of cells where we do see that consistently, depending on the stimulus. And this kind of dependency to context was unexpected compared to what we know before in the retina. So polarity is context dependent. And of course, the next question there for us was, okay, so can we find a model that kind of explain what we saw here? And, I don't want to actually go into details of how we did that, but I just want to give you an intuition of what we have done so far. So essentially, imagine that we think about the stimulus response function. So of course, the stimulus space has a very large number of dimensions, but just for the sake of making my representation easier, I just put like one dimension here for the stimulus. And the response, let's say, is kind of the number of spikes, so like firing rate. Okay, so you have a complex nonlinear function here. And what we've been doing essentially is we pick 2 points as the input stimulus here, so 2 images. And then we explored the neighborhood around it, okay? And by doing this kind of, local spike trigger average, what we've been doing essentially is to find a linear approximation of the function, of the complex function here. And, if you're a theorist, which I guess a lot of people in that room are more or less, this should scream derivative or gradient. Essentially, you can show that here, what we're computing, the slope of this is basically the derivative of the function. And so if you generalize to the case of more than one dimension for the stimulus, what we're computing is essentially the gradient of the stimulus response function. And so, I guess most of you have already computed some gradient at some point, either in your studies or in your career. Here we're doing that, but not with equations. We're just doing that empirically. And if you have that interpretation in mind, there's immediately some, some things that actually follow from that. And the first one is that, what we have found with these local spike trigger average is that it's actually changing a lot with the stimulus. And this, by definition, cannot be explained by linear function, because in a linear function, as you probably remember, the gradient of a linear function is constant. So if we see major changes, it means that a linear function cannot explain what we see. And so basically, this method is a way to somewhat tap into the nonlinear part of the retinal processing. And so to confirm this, we actually just try to find models that explain what we see. And not surprisingly, if you use a more or less a linear model, so a filter followed by one nonlinearity, so-called LN model, well, so here these are like just basically one cell. Every line corresponds to a different image as a reference. And then the first column, the second column here corresponds to the empirically measured local receptive field, local spike trigger average. And this is the prediction of the linear model. And as you see, it's constant, as the theory says. And of course, it completely misses this local spike trigger average. But we did find basically a kind of a small deep network, sort of a two-layer network with several convolutional kernels that could actually predict reasonably well, at least in shape and position and polarity, what these spike trigger average, these local spike trigger average are about. And so, like basically we learned this convolutional neural network on a different disjoint set of responses to another set of natural images that are not perturbed and are not the same as the one we are perturbing. And still, yet, the model was able to generalize, and not only to generalize and predict the response to these reference images, but also to predict reasonably well these local receptive field. And we obtained this prediction just by computing the gradient again. So we've done that. I don't want to go into details of that model in the interest of time. But also, I should mention the fact that some of my colleagues have done later another type of model based on Gaussian process, where they also show that you can actually predict that very well if you have something, a nonlinear model. So basically, this on-off polarity is context dependent, depends on the natural image you start from. And you can have, nonlinear models that predict this finding. Problem is, this learning convolutional neural networks to predict the response of sensory neurons and of visual neurons. This has been done before. There are several works, including in the retina. The issue there is, of course, they are very hard to interpret because they have a lot of parameters. So we were thinking, okay, so how can we somewhat find a more, at least in some cases, some relation between this image and the the local STA? Because I mean, okay, we could just stop there and say, hey, everything is context dependent, and you just need to take into account a lot of nonlinearities, and maybe you can get away with that kind of explanation in the cortex, but at least in the retina, we had to try to find something, a simpler explanation. Okay, so to do that, we somewhat went back to what we discovered so far. So here, what we're saying is that essentially, depending on the image you start from, what you have to do to increase the find rate is very different. If you start from this image, you have to increase light in this area. If you start from this image, you have to decrease light in this area, etc., etc. And so if you think about it in the more abstract way, it means that if you think about the stimulus space, you start each reference image will be one of these black points, and the arrow correspond to the these local spike trigger average. Basically, what this is telling you is that these arrows are going in different directions. So somehow, every time you're in different point, you should go in different directions. And again, like of course, if you have the gradient interpretation in mind, this sounds very natural. These arrows tells you where you should go if you want to increase the response. So really, the question we were asking is, okay, is there any kind of, is it totally disorder, or is there some sort of order behind all this context dependency? And to somewhat address this issue, we basically took inspiration from a mathematical technique called active subspace method. Long story short, it kind of holds in one line. What you can do is that you can just basically, so since we have this model that seems to reproduce very well this local receptive field, we could generate this pair of images and local receptive field for many different images. just by prediction of the CNN we learned before. Okay, and then what we can do is basically just like take all these ensemble of spike trigger average, treat them as vectors, and then take and do PCA on them. So basically, if you think about it, what you are doing is that we're checking the, the gradient. We're computing the gradient of, if f is the stimulus response function, we're taking the gradient, and we're doing the PCA over the, we're doing computing the covariance over the possible stimuli, and then doing PCA on it. And when we did that, we found that in fact, the first two eigenvector of this covariance matrix explain 80 to 90% of variance in many cases. Okay, so this is good because basically it means that you can project everything in 2 dimensions, and you will somehow retain most of the variance of this gradient. Okay, so this is a space where you can somewhat see what's going on more clearly, because I visualize things better in 2D, at least as far as I'm concerned. So this is what we did. And so now this is the projection of this in the 2 principal components that I should just show you. By the way, so this is all published in the paper I referred to earlier, if you want to really get the details of this. Okay, so in that space, one image again will be 1 point, and then the arrow would tell you where you should go if you want to increase the response of that cell. So now if you take other pairs of image and the corresponding local receptive field, still for the same cell, you see that they are positioned somewhere else, and as I was saying before, they are pointed to different directions. Okay, but as I said, we now have a model, so we can generate a lot of these pairs and see what it looks like. Okay, so we did that. We tried a lot of different points in this space and see where was the arrow, and we obtained this. So this is for one cell. So for one cell, you see that in fact there is a lot of order in this context dependency. Basically, all these arrows are centrifugal. They're trying to get away from that point. So basically, what this is telling you is, like whatever you do, wherever you are, if you want to increase the response of the cell, you need to somewhat get away from that point. And that point actually corresponds to a full gray, a uniform gray. So essentially, what this is telling you is, if you have something dark inside your receptive field, like here, well, you should get even darker because you want to get away from the gray. But if you have something bright in your receptive field, then what you should do is to actually increase light to get even brighter, to get away from the gray. Okay, and so basically, looking at this, you're like, okay, so in some way, these cells are very peculiar. They don't care about light increase or decrease. They just want to, get away from the gray. So in some way, what they really want to do is to compute a distance from where they are to, they want to increase contrast. So they want to compute a distance between from their area to this origin point. So in other words, inside their receptive field, they just want to take the, some sort of a variance of the luminance. So we thought, okay, so actually this is something easier to understand. Computing most of the cell we know about, I've been described as computing the average luminance inside the receptive field, but here these cells actually seem to compute somewhat the variance of light intensity inside the receptive field. So we thought, all right, so actually, if we approximate our complex model with just that, it's just basically variance of light intensity inside the receptive field, can we at least qualitatively predict that kind of our vector field? And the answer is yes, actually. So now if you do that with this kind of what we call local spatial contrast, this was also a term that was used by Tim Gollish lab. So basically, again, this is like, the variance inside the receptive field of the light intensity. Actually, it predicts qualitatively the same shape for what the gradient and all this should look like. Okay. So in fact, behind these, complex models, there is something relatively simple that you can still extract. Something nonlinear, but still relatively simple, that can explain at least qualitatively what we saw here. Just a side note on this. You may have heard of like recent work in the retina, in V1 also, where people talked about the maximally exciting image, starting with like Walker et al. From Andreas Toller's lab. And so their idea is to say, okay, so within a certain search space, like for example, this circle, I'm going to try to find the stimulus that evoke the largest response. Okay? And so you obtain, so they do that with a combination of machine learning and online experiments. It's very cool and very elegant. So what they find is usually like, 1 point corresponding to one receptive field. Okay, one shape. Well, so if you look at this and you're like, okay, so if within that circle I was looking for the strongest response, I would find 1 point, possibly this one. And actually, if you look at this one, it would correspond to something very close to that. So basically, it would look like an off-like receptive field. And if you, jump to the conclusion, I say, okay, so actually what this cell really prefers is a light decrease. Well, basically, this broader picture that we have here tells you that you actually have missed something, okay? So basically, I think if you don't know about MEI, you probably don't care. But if you have heard about it, about maximally exciting image, I just want to say, this is great, but given that kind of picture, you realize it's only giving you part of the story and doesn't tell you exactly what your cell is selective to. Okay. So to wrap up this part, what we have found so far is that, with natural images, in fact, this kind of classical on-off selectivity of ganglion cell can be more context dependent than originally thought. And you can find, models to predict that. And thanks to the model, you can actually find that in fact, there is a relatively simple explanation behind it. I mean, still novel explanation, but still relatively comprehensible and low dimension. And actually, we use that to actually make more prediction and go back to the experiment and do more tests and actually confirm this test. And this is in the paper I referred to earlier. Okay, so now from there, we're like, okay, so these kind of perturbations are somewhat interesting to uncover some of the processing performed by the retina that we haven't thought about before. Are there other places where we can apply that? And of course, everything I said so far was just like purely static flashes, flash of natural images. So we thought, okay, so what can we add time on this? And so, the next question we had was actually, what about, moving texture? So, when you look at the literature, about how the retina is encoding moving objects, it's kind of divided between like the where and the what. So the where is like, okay, so where is the object? And here you have a lot of very nice work about directional selectivity, of course, which like where is the object heading at, about motion anticipation, etc. On the what, people have actually asked about texture encoding and all these things like spatial integration and all that. But if you look closely, a lot of these work are done with like static stimuli, like flash stimuli. And in fact, when you try to do the two together, like asking what is the texture on a moving object, there's not that many works that actually have addressed that. So in some way, the kind of question we're asking is, okay, in a moving object, does the texture matter for the ganglion cells? So imagine just as a Gedanken experiment that you have, a moving bar like this with a uniform texture, like this gray texture. And now you just repeat the same stimulus, but now you replace this gray texture with like something else. Here, this is elephant skin. Somehow, again, this is purely just to set up the whole thing. We didn't do that experiment. This is not real experimental data. If your ganglion cell is responding the same way to both, then somehow it seems that probably this ganglion cell, at least alone, does not represent the texture. So we kind of set up to say, on the other end, of course, if you see something different, then so probably this cell has something to say about the texture inside the moving object. So we kind of felt like we should go there, but of course, this kind of elephant skin is very complicated. So we'd actually tried something simpler. So we just took like basically a bar of a certain contrast, and then we increase the contrast, but only, sorry, it's a little bug of the display here, either only in the leading edge, like here, or at the trailing edge here. So then at the border that is somewhat starting and the other one ending the bar. So if you just take a luminance profile, this is over space, and the arrow indicates the direction of motion, then so this is the same thing, but just represented by taking a slice in this direction. So you have the bar here. Here you're kind of increasing in light intensity in this part, and here you're increasing light intensity in that part. And that's simple enough. We can just display that to the retina and see how ganglion cells respond to it. So again, we have these 4 stimuli, the 3 ones that I presented, and also control with the high contrast here, where everything has high contrast. Okay, so this is X is space and Y is the luminance. Okay. And so I'll just show you for starters, just 2 cells which are responding qualitatively differently to these 4 stimuli. So first, let's look at how they respond to the bar. Okay, so this is the low contrast bar and this is the high contrast bar, and not surprisingly, they have a stronger response. This is fine rate over time. They have a stronger response to the high contrast bar compared to the low contrast bar. Okay, so far, so good, very unsurprising. Now, what's more interesting, and when you start to look at, what happens when you either increase the contrast only in the leading edge or only at the trailing edge. If you only increase at the leading edge, well, so first of all, okay, here, so if you look already, the difference between the orange and the red here is very small. Here it's also, it's a bit more significant. But now let's look at what happens with the green one. If you look at the green one now, okay, for the cell number 2, it triggers a very different response. So cell number 2 seems to be able to somewhat distinguish these 4 different stimuli. But if you look at cell number 1, it seems that cell number 1 is somehow putting in the same bag this stimulus and that stimulus, and also somewhat responding very similarly to this stimulus and that stimulus. And so looking at this and looking at how different they are, it feels like basically this cell is somewhat ignoring what happens here, because it responds more or less the same way to this and to that, but only cares about what happens here, basically. Okay, and so basically, if this edge is kind of the same for these 2 guys, then it gets the same response, and if this edge is actually the same for these 2 guys, also gets the same response. So it seems that you can have different cells that are responding to different aspects of a moving bar. So we're kind of intrigued by this. We were aiming at a way to test that more systematically. And this is where we use this perturbative approach again. So the idea is the following. So we start from this, steadily moving bar, constant speed. And then we decided to add to this a noise again to obtain something like this. I hope you can see the movie. So basically just, a moving bar with a small perturbation on top of it. Again, we calibrated the perturbation so that it slightly changes the response, but not more. Okay. And so now we can try to analyze what is the receptive field as a function of where the bar is. So we're going to repeat the same bar passing by at constant speed, but every time with a different pattern of perturbation, so that we obtain, we can actually map receptive field as a function of where the bar is. Okay. So first off, to really get you accustomed to the kind of representation we have, because when you add time, everything gets more complicated. Let's say you start with a simple 1D white noise like this one. This is really the stimulus that we display to the retina before the bar, okay? Well, you can actually put it in space and time. So in space and time, this is space, this is time, and you can see basically at every time step, we are changing randomly this spatial pattern, okay? You can compute a classical receptive field here with 1 dimension in space and 1 in time by just taking the responses to each of these patterns and then just doing the weighted average of the response with the pattern, and then you obtain something like this kind of receptive field here. Okay, so here this axis is time, this axis is space. This is basically what you obtain by averaging, doing a weighted average between all these space-time pattern and the corresponding number of spikes that was evoked. Okay? Okay, so this is very classical. So far, this is just receptive field mapping, but I just want you to get used to this representation with 1 dimension in space and only in, of course, 1 dimension in time. Okay, and here, last thing to say is that these 2 lines are just basically the borders somehow of this classically defined receptive field, and they are just here a visual landmark, and I repeat them for other cases. Okay, so now on the same cell for which I computed this classically defined receptive field, now I can do my stimulus where I have the bar, and then I add this perturbation. Okay, so the bar, if you define it in space and time, so this is x and this is t, looks like this. Okay, so you see like this is the bar drifting over time. And now I'm adding to this random noise pattern to obtain this. Okay, and now if I plot it this way, you kind of see the similarity with what we were doing before. This is just still in 2 dimensions, adding a noise pattern. Okay, and now I can apply somewhat the same sort of analysis where I'm going to have many sweeps where the bar goes across the screen many times. So this is sweep 1, sweep n. It's always the same bar, but the noise is different. And of course, because the noise is different, a given cell is responding differently. Okay. And now I will, for a fixed position of the bar, I'll just, do the spike trigger average over the different sweep, okay? So doing that, I will obtain a sort of a, receptive field that depends on where the bar is, okay? One thing to say here is that, of course, the bar is at a given time, is at a given position, but of course, since it's steadily moving, in this space-time representation, the bar is kind of, like oblique like this, okay? So these two lines are the borders of the bar over time relative to the time of the spikes, okay? So here you're gonna have like one local receptive field for different time point while the bar is sweeping across the visual field, okay? Okay, so now you can just look at what these locals receptive field look like and how do they depend or not on the position of the bar. And for some cells, so again, like this vertical line here are basically just landmark on where is the normal calculated receptive field. And this tells you where is the bar at any point in time relative to the time of the spike. Okay, so here you see that, not surprisingly, you have this local receptive field that is here while the bar enters the receptive field. And while the bar is passing across the receptive field, it barely changes. I mean, slightly, but barely, okay? So in that case, this is a relatively boring cells where, you have a receptive field, but that receptive field computed with this perturbative approach here does not seem to depend on where the bar is. Now let me show you another example. So this is another cell still with these two visual landmark here and the classically defined receptive field. Okay, so now if you compute what is the receptive field at the moment where the bar starts entering the receptive field, it's there. But now as the bar is progressing, you see that the receptive field that we computed here is kind of moving along so that it always stays at the leading edge of the bar. So what does that mean? It means that if you have any change in the scene, the cell does not care while the bar is passing. If you have any change in the scene, like this perturbation, the cell does not care except if it is at the leading edge of the bar. So basically, this cell is somewhat a generalization of what I showed earlier. It only cares about changes happening at the leading edge of the bar. And if you have like changes elsewhere, it doesn't care. Even if they are inside this classically defined receptive field, it doesn't care. It only cares about this, okay? So just to, this visualization is a bit complex, but so the idea that you should remember here is that basically, even if in principle this cell is supposed to be sensitive to this whole area, which is its receptive field, this is space again as a horizontal axis. In practice, when the bar passes by, the only changes that actually triggers a change in the response are the ones that are happening close to the edge of the bar, to the leading edge of the bar. Okay, and you can try to kind of make sense of this by just taking a time slice here. And then once you have that time slice, you can like put them together here compared to where is the bar. And basically, if you do that, you obtain this, which is somewhat a way to confirm, to have a simpler representation that tells you, okay, so while the bar is passing across the receptive field defined by these two dashed lines, what does the cell care about? And the answer, it cares about light increase close to the leading edge. Okay, so already what it seems is that this cell is extracting something more complex than initially thought. It only cares about what happens close to the leading edge. Okay. And like having this compressed representation, we could visualize a lot of cells, and we did find that different cells actually have different behavior. So you do have these cells which are like kind of following the leading edge. You have cells that are purely static; they don't change the location of the receptive field. We did find some cells where they actually followed the trailing edge, and we also did find cells that were just not responding to anything specific during the moving bar. Okay, so then the next question was, okay, so can we find a model to explain how this is possible? And well, again, you can actually very simply demonstrate that this cannot happen with linear model. You actually need more than that if you want to explain this data. And so I'm gonna again skip the details in the interest of time, but basically what we found is that this cell require basically a two-layer network where you have like two filters here that are convolutional. then a nonlinearity, then another linear pooling. And on top of this, it also requires a feedback that takes the form of a gain control to somewhat normalize the responses over time, if you want. And when you do have this, this is like, data, local receptive field moving across while the bar is moving. You kind of predict, you can somewhat qualitatively get something similar with the model. Okay, you can predict reasonably well how the local STA is moving over time. On top of this, we actually found that the same architecture could predict both cases, the case where this local receptive field is moving and the case where it's not moving. Okay, and interestingly, what we found is that in fact we can go from this, following receptive field to this static receptive field simply by changing this, the threshold of this dynamic nonlinearity. So basically, we did some sort of in silico experiment where we changed the subunit threshold. We noticed that basically for the following cases, usually this threshold was high, while for the static it was low. And so basically what we did is that we found that not changing anything else, just changing this threshold, we could actually switch from, a following cell to a static cell. So really the key component of the model here, I think, is really the threshold in this intermediate layer, this rectification in the intermediate layers. Okay, now, since we have this model, we can somewhat start to think about, okay, what would the whole population of this cell would look like? And so can that tell us something about the way they are representing texture? Okay, and so to to do that, what we did here is that we just display. We did simulation where we take the model. First, we imagine a stimulus which looks like this. You will see in a minute why. And now we say, all right, so let's say that every pixel in that has basically a neuron that is exactly the model I showed you before, but where the receptive field is centered only on that point. Okay, and then I have another one here, etc., etc. I have one for every part of the image of the visual field. Okay, and here what we're showing would be the activity map. Okay, so basically this is the activity of every neuron in every pixel. Okay, so if the cell only care about the leading edge, then what you should see is basically this. So you should see something where your neurons somewhat will respond to that and will have different response depending on the content of the leading edge, but they will somewhat not really care about what happens next. Okay, so you should see this pattern appearing again. If they care, for example, about the middle, then you should see only the middle appearing, while everything somewhat is similar there. And so we did that. Skip that. And this is exactly what we find. So here, this is the same stimulus I was showing to you. And for this kind of leading edge following cells, we did find that indeed there was a substantial difference between this and that. So they were really just responding differently to what happens in the very leading edge of the bar. So the bar is moving from left to right here, but they didn't seem to care so much. Their activity wasn't able to discriminate what happens in the trailing edge. While we did find also some cells that were more sensitive to the middle, and also some cells that were sensitive to what happens in the trailing edge, which correspond to what we saw with the local spectral average. Okay, and actually we could just quantify this with a D prime. They were really like with that kind of cell, you can discriminate what happens in the leading edge, but not anything else. And so I think basically what's happening here is that these different cells correspond to different cell type in the retina. Each of them actually form a mosaic in the visual space. And so what we think is happening here is that within the retina already, you have different parallel representation that are somewhat give you access to different pieces of information about the moving object. Some about the leading edge, some about what happens in general, and some about the trailing edge. And so these different parallel representation, they are not sufficient if you want to do motion segmentation, but they might just be like the first steps towards this. And I think they might just be very useful if you want to pre-process your image to start to segment objects, moving objects. Okay. So and you couldn't find this without all this perturbative approach that we've done and modeling we've done. So okay, so just to conclude on all this, on this general approach, I think there's this tendency in sensory processing these days to say, all right, let's record, the response of many neurons to a bunch of complex natural movies, and then we're gonna just throw this into complex machine learning models. And then, of course, once we have this, we can just explain everything, and that's going to be great. And what I'm trying to argue here is, let's say, a more complete approach where, somewhat we get inspiration from theory to design our experiments, because I would lie if I was saying that this idea of doing perturbation was not inspired by, for example, adversarial attack on these kind of things. But instead of just like trying to predict the responses and trying to optimize the R square, which is now even a competition sometimes, we can extract from these qualitative findings, like, for example, these kind of local receptive field, and the fact that they are changing of polarity, the fact that they are changing that are following the leading edge. Now you can ask your model not to optimize the R square, but to qualitatively predict these kind of changes. And once you have the model, you can start to really look at it, take a bigger picture, and find out, for example, with this vector field, what really these ganglion cells care about, what kind of feature they really extract during these complex scenes. And from that, you can really get this interpretation about spatial contrast, leading edge. And actually, this is a part that I haven't shown, but this actually can lead to new experiments. And I think this circle is very virtuous in the sense that it really gets us closer to understand what are the features that are extracted during complex visual scene stimulation by sensory neurons. And I think we still have many surprises and things to discover here. Okay, with that, I'd like to finish by thanking the people who did the work here. So this is my lab. I'm not the only PI here. There are 3 other PIs, and we have students and group meetings. We are a mix of theorists and experiments. But in particular, I want to highlight the contribution here of Thomas Buffet, Matthias Goldin, Samuel Vergely, and Baptiste Lefebvre, who are really spearheading that work. Okay, and well, of course, I have funding and all these things that to support my work. And thank you for your attention.